Um, we're going to hear from Emanuel Fonseca on Coccidioides genomes from low-incidence states, and that's going to be potentially revealing some complex migration history. So take it away. Thank you.
Hi, everyone. I'm Emanuel Fonseca. I'm postdoc at the University of Utah. I'm in Catherine Walter lab. And today I'm going to talk about the evolutionary history of cocci in those low incident states. So I guess everyone, many people already talk about that, the importance of phylogenetics and why that's important. But you probably diagnose valley fever, many of you, every day. But have you wondered about how genetically different are the strains from Arizona and Texas? Are cases in Washington or Oregon connect to local transmission, or they are important for endemic areas? Is valley fever even spread in a ways that we're not detecting? So many of these questions can be answered in a phylogenetic framework. And we have a lot of good examples from COVID to Ebola and even valley fever that we are using phylogenetic to get new knowledge. So hope everyone are familiar with phylogenetics, but phylogenetics is basically a genealogy tree. So a common approach is you go outside and sequence some samples, and from those samples you compare the genetic difference, and you build a tree showing who is related to whom. So you already have seen some of these trees, so it can give us important information, for example, about local structure, about travel association factions. So just like an ancestry test, it can tell us about where the pathogen came from, if cases are related or independent introductions, and how the pathogen is evolving and spreading. So we are very familiar with that valley fever is increasing over time. These include also those low incident states, just like Utah, Colorado, and Nevada. So, however, we don't know how cocci have spread into these new regions. And also genomic information is still not available for many of these states. So my goal here is to generate genomic information to clarify the evolutionary history of cocci in those low incidence regions, such as Utah, Colorado, and Nevada. So here, just summarize my samples. Here we sequence about 52 sequence from the Europe laboratory in the University of Utah Health System. Those are clinical isolates. So from that, 27 were from those low incidence states, 16 from Utah, 7 from Colorado, and 4 from Nevada. And we also add 22 isolate sequence from California. And we also use 136 samples that was previously published. So here is a map showing the distribution of our samples. We have most of our sample is in the Western US, but we also have samples from Venezuela, Guatemala, and Mexico. And some of these isolates were related to travel infection. So on top of these, of the genetic data, we actively prospect clinical metadata. Those were only from the Utah patients. And we did whole genome sequencing using short read technology. And our data set contains, surprisingly for me, almost 340,000 SNPs. This is I'm very new in cocci, but my experience, that's a lot of data. And those are only for intergenic regions. Here, I exclude exonic, synonymous, non-synonymous. So it's a lot of data. So it shows like there is a lot of genetic variation in those species. So the first thing we did was to ask what species are circulating in low incidence states. So this is a plot showing in the y-axis the number of genomes, and the x-axis is the locality. So here you can see those low incidence states. Mostly we find C. posadaceae. However, in Utah, we found both species. But thanks to our clinical metadata, we know that that patient that was infected with C. imids had a history of travel to Southern California. So probably this is a travel-associated infection. So in terms of phylogenetic relationship, as you can see here, given the data was easier to recover that break between the two species, they are very divergent. So if you look, for example, here for C. posadaceae, and if we put into spatial context our sample, we see there's a lot of going on. What I want to show is very difficult to see any structure looking at this plot, but I want to say, for example, here, we have some isolates from Utah, and they land in different parts of the tree. This suggests that those samples have different history. And here for C. imids, we only have one patient in Utah, and as I said, that was probably associated to travel. So I know that's very hard to see structure in that plot. So what I did was to take phylogenetic distance for all the samples and try to summarize this in a PCA. So here we have a PCA for C. posadaceae based on phylogenetic distance. So it shows that maybe state is not a good proxy for genetic structure. So we have a lot of overlap showing that, for example, within Arizona, we have multiple lineages. And same for C. imids. However, if you do a algorithm-based framework where here is no prior information about location, this was the one like showed before. I test for multiple number of clusters, range from 2 to 15, and we got that our optimal number of clusters was equal to 6. So here we see the division between the two species, Posadaceae and imids, and each bar in this plot is a sample, and in the y-axis, the admixture coefficient show the proportion of the genome of that sample that comes from different population. So in this case, we found four genetic clusters in Posadaceae and only two clusters in imids. And here is our low incidence state samples, and we see that they cluster in different genetic groups. This also support the idea that multiple invasions into those regions. So again, I did a similar plot, but now coloring based on this new group. And now we can see that we have like those genetic clusters. We can see clearly some divisions. And same for C. imids. One thing that we did too was do a ancestral area reconstruction. So basically, we treat the location, in this case, the states as a trait, and use the phylogenetic tree to reconstruct those optimize those characters in the tree. So if we zoom in, for example, here we have two samples from Utah, and here we see where probably was the ancestor. And here with like a high confidence, the ancestor of these two samples were likely in Utah also. But if we look further in the tree, we see that this sample came from Arizona. And we can do this for all the samples from the low incidence states. And that reveal a ongoing repeated dispersion rather than a single historical event. We found 7 introductions in Utah, 7 in Colorado, and 2 in Nevada. And no surprise, based on this analysis, the ancestral range of C. posadaceae is in Arizona, and from imids is in California. So, take-home message, our analysis reveal a complex dispersal history of coccidioides across the western US. We found multiple independent introductions of C. posadaceae in Utah, Colorado, and Nevada, indicating repeated dispersions. C. imids in Utah was likely associated with travel-related infection, and this highlights the urgent need for expanded genome surveillance. So for the future, we are in the process of adding way more samples to our data set. Also, we want to ask questions about historical demography and also how the environmental and the landscape control gene flow, and also about local adaptation, about how selection plays a role in these species. I want to thank you, all the collaborators, the University of Utah, and the fund from the Borough. Welcome. Thank you.
Fantastic talk. Thank you so much. Again, I have not been proven wrong. This makes me happy. Questions?
Okay, no one else is. Hi, perhaps I missed it. What is how much travel history do you have on the humans from whom these isolates came? You mentioned one isolate from Utah that indicated travel, but Wisconsin, et cetera. Is it important to know the travel history of the humans?
Yes, actually, this was already published and showed a lot of travel history from those samples. For what is new here, we have, for example, Utah, we only have those information for a few patients. And the one that got infected with imids had a history of travel.
So you mentioned that one because it's a new one in this work.
Yes.
Thank you.
Yeah.
Additional questions? All right, seeing none, let's thank all of our speakers for this session.